Chapter 7.
So, polymorphisms. These are when we've got abnormalities in the sequence. Okay? These sequences are caused, or mutations are caused by nutritional deficiencies. Which nutritional deficiency? I've just told you. Okay? Folic acid, zinc. So which substances are really important to all three body types? Zinc and folic acid. Good? Okay, nutrition. By pathogens. And the main pathogen there that causes these is viruses. Because viruses are strings of DNA or RNA who have the sole purpose of getting into the nucleus and changing the code. Okay? So that's their function, that's their job. Chemicals. Many chemicals are teratogenic or can damage the gene. Okay? Mycotoxins. These are fungal toxins like aflatoxin, etc. Really, really poisonous substances. Ultraviolet. And this is mainly ultraviolet B and ultraviolet C. Ultraviolet A is not a problem. That's the one that converts cholesterol into vitamin D. That's in the close range. And that's the one that the Miron glass allows through. But the ultraviolet B is the one that's damaging, and ultraviolet C, of course. Ionizing radiation. This can be from, obviously, radioactive isotopes and probably from radiation by microwaves. So in other words, the mobile phone. If you put the mobile phone, and this is quite a good demonstration really with the biggest skeptics, is put a mobile phone onto somebody and they're nice and strong. Switch it on, nice and strong, and then dial it, okay? Nice and strong, but directly they answer it, boom, they go weak, okay? Because that's when the microwave comes down and hits the person. And then you stick that on your ear and you think, oh, this is a bit warm in here. And then you swap it to the other ear, don't you, and then put it back again. And remember what I've always said, and I don't understand this totally, but the lower the pickup on here, the lower the number of bars you've got, the greater the radiation. Okay? If you've got 3 bars, you've probably got less radiation. But if you're in an area and you only got half a bar, the radiation from there is multiplied to try and amplify it. Okay? So you'll have bigger trouble with your ear. Okay? So there's no question about them. And possibly caused by thermal liability, that would be from burns and scalds, etc. Maybe mobile phones as well. So according to Bruce Ames, as many as 1/3 of mutations in a gene result in the corresponding enzyme having an increased Michaelis constant. That's a lovely biochemical word. Biochemists love naming something after themselves. Then they're made for life. This is the Christopher Astill-Smith approach. Functional genomics, functional biochemistry, Chris Astill-Smith's functional biochemistry. See, they love doing this, and they'll love making papers and love pulling papers apart, you know, from each other's work. But most important is if you've got something named after you. Okay? So the Michaelis constant, which means it's a decreased binding affinity on the enzyme for a coenzyme, in other words, the vitamin B, resulting in a lowered rate of reaction. So what this basically means is a substrate or coenzyme, and is defined as the concentration of a ligand required, that's a binding substance, to fill 1/2 of the site. About 50 human genetic diseases due to defective enzymes can be remedied or ameliorated by the administration of high doses of the corresponding vitamin coenzyme, which at least partially restores enzymatic activity. Now, that 50 is probably totally irrelevant. It's probably hundreds. You know, we know that we get good results with giving vitamins and minerals to patients. So whether you want to call them genetic diseases refers to the fact that there may be a polymorphism, which could be inherited or it could be acquired. So we won't put a figure on there now, but that's what Bruce Ames said, you know, some 5-6 years ago. Many single-point mutations, in other words, polymorphisms, in which the variant amino acid reduces coenzyme binding and thus the enzymatic activity, are likely to be remedied by raising cellular concentrations of the vitamin coenzyme. So mutations may be single. In other words, we could have a T into an A position. Okay? Or you can remove a section and put it somewhere else. Okay? A bit like happened more so in the old Word programs. You know, you'd be writing along and then you find, oh, where's that sentence gone? And it'll be down here or up there, okay? So that's a complete deletion, insertion, and rearrangements. So I call those cut and pastes, okay? So you can have single ones or you can have large sections. So any single one could be a mismatch of A to G, G to A, C to T, or T to C. Or it could have uracil into the T position. That's DUMP into TUMP. That was the first. Or there can be transversion. So you could get A into C, C into A, and so on. So that gives you your possibility, when you add uracil, of 20 different combinations. So this may lead to no detectable problem, a missed sentence somewhere along the line, or complete nonsense. Okay, so in other words, you encode for the wrong thing altogether. So to challenge, each vial of the nucleotide bases, of which there's 5, from strength to weakening over the lower abdominal area. Okay, I don't advocate putting these in the mouth. Okay, note which ones weakens. Okay, out you come. So what we do is these are, where are they? They're down there, yeah. Okay, so we'll put them on in alphabetical order. Sorry. So there's 5 of these. So we've got adenosine first of all. So we're going to put it in the lower abdominal area. And pull. So A doesn't weaken him. And then we'll put on C. Cytosine. C doesn't weaken him. G. Guanine. Pull. O. O. Okay, then we'll take that off. We'll put on thymine, because you could have more than one. And we'll put on U, which is uracil. Okay, so that's the substances we challenge for. So we found one, which is T. No, G, wasn't it? G. So guanosine. Okay. Now we go through the other 4 and find out which one strengthens. So I'm going to go and put that on the top there, and we'll find that T strengthens. Okay. So what his polymorphism is G into T. All right. Does that make sense? Do you understand that? Okay. So what weakens is the bad one, and what strengthens or negates is the good one. So in his case, he's got a G into T polymorphism. Okay. Did that make sense? Oh, good. You stay there if you will. Okay. So challenge weakening nucleotide against each of the other bases, and he's exactly the example on the board there. This will indicate the specific polymorphism. There's always an associated coenzyme for each SNP. Okay. So each SNP may be apparent to nutritional deficiency of the necessary substrates and cofactors. So we said that about acquired. So those are the genetic code. Now, let's go down the chart. This is the chart on the back here. This is also in your notes, of course. Oh, this is the notes. So what we've got is down here. So he's got a G into T. We all agreed there? Okay. We go along here. This encodes for pyridoxal 5-phosphate. So he may need more pyridoxine, or he may be one of those who actually needs the pyridoxal 5-phosphate. Okay? Now, we know that the function of B6 is decarboxylation. So we look for other enzymes which decarboxylate, and transamination. We talked about those things this morning, common things with B6 deficiency, you know, things dropping to bits. Now, we know that that enzyme is present in red peppers. Okay? So next time you have anything, put red peppers in, okay? But it's in the seed. That's the bad news. And of course, if you cook the seed, you destroy the enzyme. So you've got to eat them raw. So that. Now, there are certain, remember, toxins, certain toxic metals, certain things that can damage the DNA to cause this in the first place. So we won't let him see this, so turn your head the other way, that's it, what the illness might be. Now this could be something he's acquired, or it could be that he's inherited it. And the first person to realize this, of course, was Hahnemann, the homeopathic founder, who in the early 1800s said, there's diseases which we inherit. He didn't know anything about genes at the time, but he was the first person to realize there were factors which we can inherit. And those factors which we inherit were from diseases. And the big diseases at the time were syphilis, gonorrhea, tuberculosis, and what's the other one? So it was syphilinum, smallpox. That's right, smallpox. So it was syphilinum, he called it, medarinum, thuja, and tuberculinum. Okay, so those are the big sort of diseases. And they could potentially damage the gene. And they can damage the gene for generations. You know, they say a touch of this, or syphilis, can last 7 generations. Okay? So your only hope is to make sure that you get your children to marry somebody who doesn't have that. So it's really, really important when your children bring their boyfriend, girlfriend home and they look serious, get out your 5 bases and let's do a gene test on them, okay? And then gently persuade them or say, well, you actually need to take a bit more P5P, I think, okay? So this is where you'd inherited it. I'd like to tell the story just at this moment, and I've told this story many times if you've heard it before. When I was a new graduate, we had somebody, chiropractor in the clinic in St Hall, who used to do pendulum work. And there's a bit of voodoo in those days when people used pendulums. And I plucked up courage. I said, Bruce, would you do an analysis on me? And he said, yeah. He took a blood spot, that's right. And a week or so later, he said, oh, we've done a test for you. And I said, yeah, oh, what came up? He said, oh, gonorrhea. And I said, oh. He said, but it's all right, it's inherited. It's not you, it's inherited in your genes. So he said, you just need to take some Meridian, you know, and that'll sort it out for you. And so we didn't know anything about coenzymes, we didn't know nothing about vitamins really in those days. And so that Sunday, or the next Sunday, I went to the Sunday lunch as you do with your parents from time to time. And I'd sort of broached this subject about, I'd had this test done, and that gonorrhea had shown up, and it was an inherited factor. And my mother looked at my father and said, it was him. Now, the problem is that he had heart disease, and he had all the classic things that we talked about with P5P deficiency. I didn't know that in the days when he passed over. We probably nowadays, we would have said you need some P5P. He had heart disease, he had obviously had homocysteine, high levels of homocysteine, and his father also, okay, had it. He died of a heart attack at 56, the grandfather, and Dad passed away at 71. Now, it's very important, of course, if you've got that in your family, that you see, ooh, I'm going to do something about that. And I know that I got a lot of mouth ulcers as a teenager. And I know when I start to go low in B6 or pyridoxal 5-phosphate. So I begin to see the symptoms. Now I think, no, I'm just going to take that regularly. It's a lot easier than saying, oh, I'll wait for the skin to start dropping off and other things to drop off, and then I'll start taking it. You know, the answer is do it now. You know, start, find out what your jungle drops, as they call them, are, what exactly vitamins, minerals, oils and things that you need to take, and take them on a regular basis. You don't need much, but it's easier to do that than to take it when you need it and then expect to get back. So this does not mean that he has this or that this is in the family. This is an example, because it could be a toxic metal that's created this, it could be a chemical, it could be radiation and so on. But we do know that when this shows up, it encodes for more pyridoxal 5-phosphate. And on studies that they've done of soldiers that they discovered, French soldiers, evacuating from Moscow, mass graves in Poland have been discovered of French soldiers who, on the withdrawal from Moscow from the Napoleonic War, every single soldier had syphilis. Every single soldier. So syphilis must have been completely rampant, you know, in those days. This is only, 1812, of course, wasn't it? And, you know, that's without gonorrhea and those who had tuberculosis. So the gene pool wasn't that good, you know, when you think about it. So it's not surprising, you know, it's only 200 years, and we're still sort of getting over this. And so make sure that we try and ameliorate this the best way we can, that if you encode for pyridoxal 5-phosphate, take it. Don't not take it, because you're not doing a favour to your offspring. And if they've got a polymorphism, make sure they take something regularly so they don't give it to them. And it has been shown that you can get rid of these polymorphisms, providing you take it before you have the offspring. Okay? If you've had your offspring, then it's too late. But make sure you test your children. You know, you should do this with your children. Test them, see what nutrition they need, and get them onto it now, and try and persuade them that it's a good thing to prevent problems from later on. Okay, so assessing the optimal. Oh yes, this is the last bit we want to do with you. Okay, so what we want to do is assess with the weakening nucleotide base on. Have I got that one here? That's the G. Okay, with the weakening nucleotide base on. challenged with the appropriate coenzyme, it should strengthen. So let's go to our nutrition kit, and what we'll do is we'll get out pyridoxine and pyridoxal 5-phosphate. Pyridoxine is the basic vitamin B6, so-called inert, and is of plant origin. Pyridoxal is another form of B6, which is of animal origin. So pyridoxal 5-phosphate is what is made in our cells, because we're animals, but we eat our pyridoxine from plants. And the major plant, the best source of B6 is brown rice. Okay, good. And again, you must drink the water. No, you don't drink the water, you boil the rice till it's dry, okay? If you wash it under the tap or you don't strain the water out, you've probably lost most of your B6. So he's got his G on with the weakening nucleotide based on the patient challenged with the appropriate coenzyme. Now we're going to put on B6 or pyridoxine. Good, and he strengthens. And then we'll put pyridoxal 5-phosphate on, which is the activated form. Excellent. Okay, so he strengthens to both, as you'd imagine he would. But which one do we give him? This is the $100 question. Can he convert pyridoxine into pyridoxal 5-phosphate? Or are we wasting his money by getting him to buy P5P instead of pyridoxine? So let's cross therapy localized now to GV20. That's the hypothalamic point on the top of the head. So what we've got now on there is pyridoxal 5-phosphate. GV20 is the acupuncture point for the hypothalamus, which is the regulator of all the body hormones and brain chemicals. So we're asking, in relationship to pyridoxal 5-phosphate, how do you like that? Okay, and he's saying, love it, nice, stay strong. Okay. Then we'll do conception vessel 22, which is right on the top of the manubrial sternum there, which is the point for the corpus callosum. So this tests the left and the right brain. How do you like that? Love it. And then I like to do the pituitary gland as well, and stick the tongue on the roof of your mouth on the cruciate suture, in other words, governor vessel 28. Fabulous, he says. Okay, so this is good. So what he's got there is he fulfills all the requirements for pyridoxal 5-phosphate. So now let's do the same thing with pyridoxine. Okay, so in other words, wait, let me just test. So let's put you into, right now go. Go here. Go on the roof of your mouth. Okay, so he definitely is a P5P person and not a pyridoxine. Okay, so he told me he has difficulty. Everybody tells him, you know, how to help him with his health-wise, and you find out what it is, and he feels better for a few days, and then he goes back again, doesn't it? And that's probably why, is because you're one of the few. You're one of the 5%. You can make all the difference in the world. So what I need is a bottle of P5P from over there. Clive, a bottle of P5P. Solidor? Oh, well, give me a sample bit. It doesn't matter, as long as I got some to just a muscle test. So I want to just remind you how to do the test. Remember, I'm just looking, in this case, in isolation. We're not looking at the whole picture. Okay, we're just looking at the situation. We'll come back to how you look at a patient in a minute, or a bit later on. So what we want to do, you hold that, is what we want to do is to be able to assess how much you need. Thanks. So these are beautiful little bottles. So we take up a squirt. So you might want to do it as drops. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay? Okay, so we just about squeezed 10. So 10 is a squirt of that. We've had difficulty, as you know, being able to get graduated pipettes. We prided ourselves on the fact we did have graduated pipettes. And then the guy, which appears there's only one company in England at least who make graduated pipettes, went and died. So he clearly was a red, and he got high homocysteine. He should have taken some B6. So what's that, 2? Okay, this is not going to be a couple drops. Okay, if you ask him how long he hasn't felt well, or tip-top, is how long? Long time, many years. So it's not going to be a couple drops. It's going to be a large dose. Okay, and that, remember, is separate from what he needs as, he's a red, I believe, so what he needs as a red person, he needs this in addition. So that's 5. 5 squirts, lovely. So how often does he need to take it? 3 times a day, she says. She's a big prescriber. A good customer. Right, okay, so stomach time, yes. Circulation sex time, which is supper time, nah. Small intestine time, nah. So just for breakfast, okay? Usually P5P we don't citrate it, and in fact it doesn't usually like citrate, which is why I don't suggest with P5P you take it in orange juice. Take it in water, but it doesn't go well with citrate for some reason. So our activated vitamins, our P5P, methylcobalamin, adenosylcobalamin, riboflavin 5-phosphate, don't put them into fruit juice, okay? And we don't citrify them, okay? So they're active just as they are. Just pop a bit of water with them, okay? Yeah. Now, okay, you pop back. So remember we looked at this in isolation. When we come to put it all together, we might remember that this is a factor with him, but we wouldn't just send him off and prescribe P5P on its own. We would want to see what he needs generally. Now, beauty of these bottles is if you don't put it in the water, you can pop it back in the bottle. Give it to the next patient. Okay, that way you don't waste your test kit. You'll waste a minimum amount in the bottom there, but not much. If I don't replace everything, Clive, is very, very cross, isn't he? What? Give it to the client? No, why didn't I give it to the client?
Well, because you stopped you from testing further.
Yeah, it would stop me from testing anything further, but most important, it's a waste of time, isn't it? It's the wrong time. Stomach time is 8 o'clock till 10 o'clock at the moment, but remember in a couple of weeks' time, the clocks go back. It's going to be 7 o'clock till 9 o'clock, which is a nicer time to take it. So he fulfilled the requirements, GV20, conception vessel 22, and GV28. He fulfilled all the requirements that he actually needs the activated form. That's how you do it. So examples of activated vitamins would be those. So here we've got a chart, and this chart again is available over here, of how the vitamins are activated. I love doing this. These are activated vitamins. Alan was asking me, have you done one of vitamin D? Yes, I have, but vitamin D is not a coenzyme. It's a vitamin. So here we see simple ones like B12. How do we make methylcobalamin? We added it on here. How do we make all the folates? Here's tetrahydrobiopterin. Here's biotin, vitamin C, SAM, that was the one we did this morning. And here's how you make CoQ10, the nutrients that you need, and so on. So it was nice putting it together. So you want to see if he had strengthened to pyridoxine, all right, and not to pyridoxal 5-phosphate, the USO, it would have meant he needs B6 and the cofactors to get it to work. Okay? Now most of the time these are zinc and magnesium. Okay? But if you have the chart, which I strongly suggest you do, what you would do is, all right, there we are. You would look at the chart here and say, well, he's strengthening to here. How do we get him to make this? And we look here, it's ATP, which needs magnesium and zinc, and here it needs B2. So you look at the cofactors that allow him to make it. So you may be able to convert pyridoxine, and he could be just B6 deficient, but in his case he's not just B6 deficient, he actually is deficient in the activated form. Now it doesn't mean he doesn't make any, because he would have been dead years ago. But here we've got a slow polymorphism where every year he gets lower and lower in his tissues. And this is the story, isn't it? Every year, and nothing really brings him back. Okay, so he might be here, and next seminar he comes back next October, he could be here, and then here. But we saved him. He's saved. So massive doses of pyridoxine. So that'll be a very high dose. That is certainly not the RDA. The RDA is 1 drop. That's how I measure them. So he needs 5, which is at least 50 drops. Okay. So cross TL to GV1 weakens the muscle, then the body wants the patient to synthesize their own enzyme. Good. Okay, now that's the hardcore stuff. So when would you test a person for that? Really when a person's strength is indicative they need a nutrient. Okay, so when the time comes in the patient protocol and you're looking and nutrition comes up, which I usually do with the eye position to the left and up, you think, they need this. Let's check a person through for the vitamins, let's check them through for the minerals, and let's check them through for coenzymes and so on. And when a coenzyme comes up, it means you probably got a SNP. Okay? If a coenzyme doesn't come up, then you probably haven't got to bother about going down that pathway. But if it does come up, a coenzyme comes up, then I would look for a SNP. In other words, verify which one it is, put the five bases on, and if you get the code comes up there, you just go for your notes there. If you do it long enough, you begin to actually remember some of these. You know, some of the common ones, like G into T I always remember, A into C, C into A into C and C into A are the ones for the B12s and so on. So there's common ones that you can remember. You're probably more likely to remember the diseases with the patient. But remember, don't go to the disease and say you've got that, okay? Because that doesn't go down well with them. And also they probably haven't got it, but they may have the coding for it inherited, you know, from a miasm or acquired. So that's the genetic parts. What time are we going to break for afternoon tea? About 3:30. Okay. Epigenetics is, epi means above, as you know now. Epidermis means the outer layer of the skin. It's above the skin. So epi means above. So epigenetics is things that can influence the gene. Okay. So the genetics, the problem with genetics we know is the gene can be affected by radiation, viruses, etc., and can create polymorphisms and damage. But epi means things that we can regulate. So things which are useful for the cell and things which are harmful. So epi means studying about toxins, epigenetics, things that can damage the gene. We know our toxic metals, our viruses, are the foods we eat, our deficiencies. We know that other things influence can have. Oxygen, for instance, creates free radical reactions, so this is important, hypoxia. We know chemicals, a lot of chemicals can damage the genes. And when I started reading about what epigenetics is about, the influence on the gene caused by nutritional deficiencies, by toxins, by the food we eat, and most importantly by the thinking that we do, or does our chemistry, I thought, well, that's what I do. Isn't that what I do? You know, I've been looking for a name of what I do because I got stuck with functional biochemistry because I taught biochemistry. But now looking at more holistically, we want something that incorporates the mechanics in there and most importantly there, the emotions part. And so epigenetics really is something that encompasses much more of the work that we now do or we now teach. So we don't call it functional biochemistry. The biochemistry keeps us just to the biochemistry side. Okay, so I like the word epigenetics, and I liked it so much I thought I'd form a company. And amazingly, there's a man from Remington Shaver who used to say that, didn't he? Do you remember? I liked the shaver so much I formed a company. Okay. And amazingly, the name was very free. You know, somebody from Australia at the conference said, "Oh, you know, you couldn't possibly form a company called Epigenetics because it's a sacred name like genetics. You couldn't form a company." I said, "But I've got one." He said, "Have you?" Yeah. So, epigenetics. So here we see a picture of the proteins surrounding. You see this protein surrounding called a histone, surrounding the DNA, and these little parts or tails which are attached to the histone protein, and this is where the methyl group or acetyl group gets attached to, and that either slackens the protein or closes it. Okay, so the big beauty of that is it helps to open the sleeve up, but we don't want it opened up too much, otherwise you might expose a bit too much information. Okay. So that's what that said. Okay. Okay, methylation. Now, we talked briefly this morning about methylation and how important that was, and particularly in relationship to keeping the lead on our genes. But methylation is also important for converting phosphatidyl ethanolamine to phosphatidylcholine. It's involved with noradrenaline to adrenaline. It's involved in the metabolism or breaking down of dopamine, noradrenaline, and serotonin. It's involved with the metabolism of estrogens and testosterone. It's involved with DNA methyltransferase, methyl caps we mentioned, etc., etc. So it's involved with the synthesis of myelin. So methylation itself is a really important pathway which you should really understand. And looking back at those pictures of it, with the production of SAM from methionine, is quite easy to work from. So this is the big chart, and we do this as a chart if you want this. And I believe, I'm not sure whether this one's on the website, but we do all the charts that we do, the laminates, are available free of charge on the website. Okay? The only thing is that you don't get them come downloaded, laminated. Okay, so if you print it off, you want to make a big one, do an A3, and take it round to somewhere who will laminate it. Okay? Or you can buy a laminated one from us in the first place, whichever way you want. But this tells you the whole story if you can understand it. Okay? So this is the bit we looked at this morning. Methionine going around to homocysteine, going on to cysteine sulfate for the sulfate oxygen pathway. Okay? This around here tells us about the folates, why the folates become methylated here to form methylene tetrahydrofolate, methyl tetrahydrofolate, which then donates its methyl group to B12 to cause methyl cobalamin that recycles it. So it's a beautiful chart. It's so good, you know, I made it. And I'm about the only person who understands it. So, okay, now let's get down to the other aspects of the work today. Now, first of all, the patient comes in with a bad hip. We talked about that, you know, and they've got a weak psoas. Okay? And you follow that back and you say, yes, you've got a weak psoas, and this is related to the kidney, and the kidney is related to the kidney meridian. So the person strengthens the kidney meridian, and we find that they need a few nutrients to try and spice them up. We know that the kidney meridian is associated with high levels of serotonin. And as we've seen, serotonin is metabolized by methylation, and so they may show that they need some methyl donors, etc., etc. So there we fix the chemistry, but there's an emotion involved with the kidney meridian. The kidney meridian is involved with anxiety and fear. Okay? Now these emotions were first of all plotted by David Hawkins, who I have to, if you, in case you didn't know, is now the late David Hawkins. He passed away a few weeks ago. So he's up there watching us do the work. Okay? Now I managed to equate his scale of consciousness of 1 to 1,000 with the different meridians. And what his scale says that up to 540 you live in the physical material world. When you achieve 540 and above, the main emotion that you express is unconditional love. And unconditional love is unconditional love for everything. Okay? Now that's an extremely difficult situation. You can have unconditional love for most things, but you know, I don't like Manchester United football team because they beat Southampton 41 years ago in the cup final. So, you know, I hold grudges still from the past. Some things are very hard to let go of. So that's not unconditional love, okay? So levels below 200 are people who have difficulty in certain respects of telling, you know, truth from falsehood, as he calls it, and are really engulfed by unconscious or negative emotions. Let's call them negative emotions. And these are things like guilt and blame, like anxiety and fear, like grief, like motivation problems, apathy, like pride and so on. When we go above 200, we come into the more positive outlooks in life, ranging up from the other different emotions of, as we'll see, up to love. So love is the top one of those, and love equates with the small intestine meridian. So we'll see the other meridians are in a particular order there. So if you could diagnose which meridian was the primary meridian with a person, you'd be able to tell what their primary emotion is, assuming that they're not above that. If they're above 540, they won't show a meridian imbalance. Yes, they're alive. Yes, they have meridians. But what's the difference? The meridians are in balance. The chi is flowing down the meridians. As you'd imagine, a person who's in perfect harmony with the world will be perfectly harmonious in their body chemistry. Okay? So this is why David Hawkins said, real healing only occurs when you achieve 540. So that's a big, stiff order for most of us. Okay? But if we can get people into harmony or coherence with their meridians or their energy fields, then they're more likely to be good in their chemistry and balanced. Okay? So this is the most we can hope to achieve, is to balance the person. Now, why are we out of balance in the first place? Okay? And the simple reason is we have a left and a right brain. Now, cows don't get out of balance. They don't worry about how much money they're paying tax. They don't worry about politics. They don't even worry about factory farming. You know, they have instincts, basic instincts that they react to, like fears and things when someone comes along and damages them or they're terrified, but they don't have thought process in the same way that we do. And the reason we have conflict is we have a left brain and a right brain which are lateralized into different functions. Now our left brain is our logical brain. This is linear, this is where we make straightforward decisions. Our right brain is our intuitive brain. This is where our thought processes come from. When we think of an original idea and we think, wow, that's fantastic. And then the left brain goes, no, you haven't got enough money. You can't do that. You can't do that. So left brain is very negative. You know, I don't like my left brain. I like to live in a world where I'm positive. So when I went to do this digital photography a few years ago, to see how you can actually see this in the face and measure it with the colours, as we'll see, the professor said, he said, good God, he said, you're right-brained. I said, oh, sorry. He said, I thought you were left-brained. So I said, why? He said, I've taken photographs of you on the internet of every trip you've done into Russia, and I've seen you from when you started 20 years ago, or 15 years ago there, coming to Russia. He said, you were left-brained. So he said, you've changed. And I said, well, I think I know I've changed, okay? I used to be logical. If you told me any of these things 10 years ago, I would have, you know, laughed at you. It had anything to do with the work we were about to do. You know, it was fixed structural work for me, fixed biochemistry. Everything was validated in Harper's biochemistry. That was who I was, you know, very rigid in my thinking. Now, sometimes I have to admit I'm wrong. I'm learning humility for the first time, okay? But I'm also listening to that intuitive side and creating, and it's a wonderful voice that talks because it's a creative voice. And now the left brain says, all right, I give in. You can have what you want, you know. You've been through all the hardship. If you want an idea, then go and do it, you know. So it's much more positive towards me. So if I come up with an idea, I then work towards creating that idea, which is nice. So my brain, or my meridian system, is much more in harmony. It's not perfect yet, but it's much more in harmony than what it used to be, okay? I was right over here on the left, and then for a while, I swung right over here. And I've told the professor, he said, you've got to get back to doing some osteopathy, Chris. I said, why? He said, it's mechanical. You've got to get that left brain working. So I do sums and things now. You know, occasionally I sort of try and stimulate the left brain. Because otherwise I find myself living in that world, and I'm sure we do. And when I started testing people for left and right brains, we found that most of you are right-brained. Okay? That's why we're healers. That's why we work in this field. If you were left-brainers, you would be medics, okay? Hospital-type medics. The more research, clinically based medicine that you do, evidence-based medicine, the more left-brained you are. Left-brainers don't create anything. They're too busy arguing about the evidence that this works. And I don't think you should do this because, you know, blah, 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 blah. This person says, I've got a great idea, but never gets it to fruition. Okay? So if you're here, you're more likely to be able to create and do it, or get somebody else to do it. So if you're a right-brainer, and we need right-brainers because they're the creativity, for God's sake, marry a left-brainer. Okay? And if you're a left-brainer, marry a right-brainer. If you're in a company, you need a right-brainer who's a creative. But they're a pain like me for administration, aren't they, Clive? Okay. You should see my desk. Judy comes into my desk, she just takes the whole of the desk and puts it in a box. And it stays there. I don't worry about it then. And then you've got to have a left brain. So I need a secretary. I had to have somebody to look after my administrative side, because I hate that side of it. But you've got to have a brain. You've got to have your administrators in the company and in a house and in a relationship. So a right brainer should really get together with a left brainer and vice versa. So if you're getting together with somebody, do the muscle tests that we're about to do. Test them for SNPs and test them for their left and the right brains. So you can see that this sort of flow that occurs is we have a thought, and that thought comes from somewhere, but we don't know where it comes from. Very difficult. The thought doesn't have language, doesn't have words, it hasn't got to that stage. It's an idea. Then we think about it and we put it into our conscious field of awareness. So we start to play with it and we put language into it. But the language is only symbolic of what the thought is about. So we then begin to think, oh yes, I could do that, I could create that. And then you start seeing somebody in the lab making it, etc. And then you begin to remember, now what was it? You know, if you talk about the dream you had, or the idea, and you talk about it and you write it down, you remember it, don't you? Otherwise you have this fascinating dream, 10 minutes later you can't even remember what it's about, and yet it was so fascinating at the time, because you haven't hardwired it. And you hardwire it into your brain, neurologically, by repetition. This is why you talk about it, and you say it, and you think about it yourself and the self and the self. And this is why when you wake up and you've had a nightmare, you don't forget the nightmare quickly, do you? Because your brain goes straight back to thinking about it, and it hardwires it in, wretchedly, okay? And this is why it's very difficult to get back to sleep when you've had a nightmare, because your brain goes straight back into thinking about it. So when you hardwire an idea in, you hardwire it, and we call that the subconscious. So the subconscious is everything you've ever experienced. Everything you've thought about, everything you've seen, tasted, touched is recorded in there, okay? And under hypnosis, this can be extracted, okay? But voluntary, it can be difficult sometimes because you haven't hardwired it enough. But under hypnosis, it can be extracted. We were just saying at lunchtime, where's Lynn? Oh, there she is. You received, sometimes you can't always rely that an email gets through. But sometimes a text or an email comes through some weeks or months later, doesn't it? Suddenly appears and you think, where's it been? And this is a good question I heard. A youngster asked me, what happens to texts when they're not arrived? And they've been created, you see, they're energy fields. They've been created, they're electronic forms of energy. They go out there and then they go around and they are still there. The world must be full of information. Absolutely full of information that's never been delivered yet. One day someone will collect all that and absorb it and clean the airways, otherwise the letterboxes will be full, the mailboxes will be too full. But information is really interesting. The more you hardwire it in, the more you can remember it. But the problem is, if you hardwire something in, which is not nice, it'll stay there. So if you're hardwiring in anxieties, fears, anger, guilt, it becomes hardwired, and you then live this, and you don't know that you're even doing it. And if you don't know that you're doing it, these negatives, it's called unconscious emotions. So unconscious are negative ones that you're not aware that you're actually doing it. Okay? So I don't know, myself, what I'm thinking about, how I'm coming into the world, but you can test me, okay? And you test it simply by finding out which is my primary meridian. So before we find out the primary meridian, we've got to know that I'm processing correctly in the first place. Okay? So we want to know where I'm coming from. Where am I coming from in my mind? Okay? Because nobody knows what the mind is. What is the mind, people say, is something which is non-linear, and therefore you can't measure it. So to a scientist, a left-brainer, it doesn't exist. It's a series of chemistry reactions in the brain. Okay? If you're depressed, you need this pill. If you're anxious, you have this pill. To a psychologist, it's all in the mind. Okay? The body doesn't matter at all. It's what you are in your mind. So the truth is sometimes a little bit of both, isn't it? It's not the left brain or the right brain, the psychologist. Sometimes it's this, sometimes it's that, sometimes it's a blend of the two. So we need to address ourselves, is the conscious thinking with this person, is it positive or negative? Is it coming forward into the world and constructive, or is it being destructive and going inwards? So according to David Hawkins' chart, people who calibrate below 200 are in the fact the world is in control of them. They've withdrawn into themselves. People above 200, up to 540, are coming forward in their lives. They're in control of their lives and doing something. Those who are above 540, well, once in a while you might meet somebody. I'm going to show you a picture of one a little bit later on. Okay, so everything must be in perfect balance or harmony within the different aspects of looking at a person. So ultimately we want the meridians, we want the neurological system, we want everything to be in balance. Now, we did this a while back of how we can enter into a patient's problems. And we used a lot of times when we used coloured acetates or coloured filters to be able to get into the body type. And I like this. This is a really good way of doing it. And it's very reliable to know what the body type or constitution of a person is. So we'll whip through those. So those body types we've talked about a lot. Most of you, I think there's a few people who may not be quite as aware of what these colours mean. So we know that people have cones in the back of their eyes which are offset to the orange-red zone, to the yellow-green and the violet-blue. But we also know that these colours, or particular colours, are wavelengths. Every colour is a wavelength of visible light, and some wavelengths we like and some we don't like. Some colours you'd never wear, would you? You'd never even look at, and other colours you say, oh, I tend to like that because that's harmonious or complementary to me. So let's get that one. So if we put the red, green and blue colours on a person, they tend to weaken to the one which is bad and stay strong to the one which is good. And they were weakened to one of these. And it took me many years to try and understand why this was. Many years ago, we did know that people who were red tended to be sensitive to wheat. That was the first thing we found out about them. And then people who weakened to blue were sensitive to lactose, to milk sugar. So I thought, oh, maybe green people are sensitive to eggs. But it wasn't, no. It wasn't that easy. Okay. And for years then, slowly bits of the jigsaw came together. We found that blue people were sensitive to mercury. Okay? So these were the people who had the problems with leaking amalgams. Whereas red people, and I was red, people were sensitive to aluminium, or aluminum, as Alan would call it. Okay? Green people were sensitive to nickel. Okay? Otherwise, we didn't find too much wrong. We knew green people were very sensitive to drugs and chemicals. They were a nightmare, but we didn't know anything more about really how it all fitted in. And it wasn't until I started to put in to looking at the toxicity with the cytochrome P450 enzymes within the toxicity box, and that we found that red people, green people and blue people were specifically deficient in cytochrome P450 family. Okay? So the 3A4, which detoxifies 70% of all known drugs, is deficient or defective in green people, which is why they're chemical nightmares. Okay, they can't tolerate certain chemicals because they don't have the ability, the liver enzymes, to detoxify in sufficient amounts. Obviously they have some, but they've drawn the short straw on the expression of that gene. Okay? Whereas a red person can eat all the foods, eat all the poisons and things that a green one can't. And similarly a blue. But blues were nightmares when it came to certain other chemicals, and most of those chemicals were in toiletries and cosmetics. And that became a blow because women were then started to weaken to the things that they put on their bodies. And if they weakened to their toiletries, that was okay, but when they started weakening to cosmetics as well, that was the end. Okay? So there's certain things which people will put on their face, and then we found that men were the same. Blue people were using shaving foam, and shaving foam is full of estrogen mimickers. The sodium lauryl sulfate is the worst one for shaving foam. And men are shockers at grooming these days. We like to look really smart for the girls, okay? So hair gels, all this, shave, all this, and it's all estrogen. You know, they're all estrogen mimickers, or most of them, okay? So we had to come up with a range of natural toiletries which didn't have estrogen contents. And sometimes even those, even though they're made of organic and pure materials, even those sometimes weaken. You know, we found somebody who was sensitive to one of the aromatherapy oils in there. You know, people can be sensitive to anything, but the greens tend to be the ones who are most sensitive to chemicals. Green people should have been born 200 years ago, okay, when the world was cleaner. Then I started to find gradually a little bit more interesting part to this. They fitted into different body types. And this was really interesting because I noticed that green people were all around Scandinavia when I teach around Scandinavia area. And we've done Norway, Sweden, Russia, around there, and Latvia. And practically everybody is green. Okay, they're all weakened to green. I thought, well, why is this? There's something about the people here, either the environment here that makes them very tall, or that they're a particular race. And we noticed they were all tall. You know, Scandinavians are rapists and pillagers, aren't they? They come over and they raid our villages and things like that. And then I started noticing that the blues, the true blues, were all small. And they came from the east. They're like the eastern people. They're shorter build, okay? So we found a measurement. We said, right, 5'5" for women. If you're below 5'5", you'll probably be blue. If you're above 5'7", 5'8", you'll probably be green. Therefore, you're red in the middle. If you're in between there, you're probably red. And give or take a few inches, okay? This is not 100% hard. You could be a few inches either way, okay, difference on there. But generally, and men, of course, we got to add an inch or two on to begin with. But there was another giveaway, not only the height to what the person's body type, but the hand. So those of you who haven't done that yet, look at your hands. Linda was an example here. Linda, you were telling me you're 5 foot, how much? 5'1.5". 5'1.5". So you would immediately say she was in the blue zone, okay, at that height. Now, look at your hand. Is the hand square? Okay, in other words, is the palm and the fingers roughly the same sort of size? Or have you got long, thin hands with long, thin fingers? Or do you have small little hands with small little fingers? Okay, as if the top digits were missing. Okay, okay, the short ones. Okay, so we all have different size hands, and you may not have ever looked at your hand in that sort of way. Is it square? Okay, and what we found is that some people have little hands. Stick your hand up. See the fingers there? Okay, little. She's had the top digits chopped off. They do it at birth in certain hospitals. Instead of wrapping up the feet to stop them growing, they chop the hands off. Okay, this does not apply to the feet. Somebody said, well, does the same thing with the size of the feet? I don't think so. No, you can have big feet in little people and vice versa. Okay, but it's the hand. It's a big square working hand. This is an ideal hand for an osteopath, chiropractor, manual therapist. Okay, you need a hand like this. Okay, a long thin hand is very nice, but it's not so good for the manual work in the sense it's going to get tired. You see the long thin hands playing pianos. That's where they belong, really.
And where would you put those? Green?
What?
The
Long thing is red. Okay.
Thyroid.
It's the thyroid. So the last bit of the jigsaw, apart from the physical body, is what makes the physical body the shape it is. Why are people different body types? And the answer is it depends upon which hormone is dominant in that person. So the thyroid people, who are the reds, are the thinner ones when their thyroid's working. When their thyroid doesn't work, they go outwards. You can never grow taller when you're over 24. Just give them maximum amount of growing time. So if you're over 24, you probably won't get taller. Ultimately, you'll probably get a bit shorter, but your body type will remain the same. Now, you might be thin, red, but when your thyroid starts to fail, you'll gain this way in a particular weight gain pattern. If you're a blue, you're dominated by your gonads. So both boys and girls will be there down, and they're shorter build in there. And as they fail, again, you get various weight gain patterns because the hormones, particularly the estrogens, get out of balance. Okay? The adrenals are the greens. We're the tall people, and we go, and drop. Okay, and when we drop, we're in serious trouble because it's very difficult to get back up again because we don't rest enough. So those are the three sort of body types. And gradually what we've done, and I'm about to, as soon as I got a little bit of time, to build a sheet, to make a sheet of each body type that you can give to patients. Because this is when you prescribe your remedies, you want something that they can go home with and say, I remember you telling me this. Now, what was it you said about me? I'm a red, and you muttered something about, should I eat takeaways in aluminium? I can't remember what it was I said. So they want it written down, no aluminium or avoid aluminium, etc., etc. So red, lean is thyroid. We won't go through all the different bits because we've done through that. See the long, thin fingers? And this one is a weight gain of a lady, and this is a thin, scrawny man. Okay, so this is a red type. Medium build, I've actually on here put 5'5" to 5'7", probably 5'6" to 5'8", but it's not written in stone because sometimes people are shorter but are one body type. Okay. Now this is the key stuff here, and this is what I want to put in an order that you can give to patients. So affinity to aluminium. Okay. In other words, the detoxification pathway for detoxing aluminium is down. It's the short straw. Okay. Homocysteine. Remember what we talked about this morning? Homocysteine. That's the need for the B6. Low methyl tetrahydrofolate, so they need folate as well. They're positive ApoE4, which is the expression of the apolipoprotein, which leads people to have high cholesterol and triglycerides. They require iodine by their thyroids, of course, and by their immune system. They're natural carnivores. In other words, they like high protein diet. They react to wheat, okay? And then you can avoid broccoli, that's the good news. And because broccoli contains cyanide compounds which inhibit the thyroid from working, and they're sensitive to sulfites. So we need to put that in a nice little package which you can give to your patient. Okay? So on the back you can have something that why you prescribe the following remedies. These are the remedies that the person needs to take for the following reasons. And let's have a look at what they are. So vitamins, we're just coming for tea. Vitamins that a red person needs. B2. Okay? Remember, this person has high homocysteine. They need to be able to metabolize this and get rid of it. And to recycle it, you need B2. B6, pyridoxine or P5P, folic acid, B12. And here this person needs inositol. Interesting. I don't know why. Something to do with, obviously, phosphatidyl inositol. But they show 2 inositol. And vitamin C, which is what I mentioned, but more so in the winter. They probably won't show to vitamin C at the moment. So red people should take those. Now, they don't need to take them individually. What we do is we mix them all together and put them into one bottle, okay, along with the minerals. So the minerals that they always show to is iodine, of course, to get the thyroid going, and magnesium and potassium iodides are the best source, bioavailable source of this. They need manganese, strengthen up the ligaments. They need molybdenum, remember why, for sulfate oxidase. They need selenium to activate T4, thyroxine into T3. They need silica to give them good, strong joints. And like everybody else, they need zinc. Okay? So what we've done is we've made a mix of all those vitamins and minerals and put them in a bottle. And we call it a red solution, a red vitamin mineral mix. And the beauty of this is it supplies everything that a red person needs. Okay? Now, there may be the odd case who needs a little bit extra than this, but generally most people don't. But sometimes people need a little bit extra, and you're one of those who needs some extra P5P, okay, over and above the ones that you standard sort of remedies there. Okay? So what we do is we mix the same amount as in every bottle of those individually. So 1 squirt of red mix will give you the same as 1 squirt of iodine, 1 squirt of zinc, and so on. So it's very concentrated. Okay, so it's a multiple liquid mix tailored to the constitution of the person. Okay, so chemistry from now on becomes very easy, you know, too easy really, and everybody turns around and says, "Oh wow," you know. And the Russians were so delighted about this because all now they need to do is to order red, green, or blue mixes. Don't have to think at all. Now we did the same thing. In fact, the first thing I did with people was to test their oils, and we found that flaxseed was very important. Flax is rather pungent, but it's more omega-3. Hemp, which is sort of blend of omega-3 and omega-6. Olive, these are the only people who should eat olive oil. And pumpkin, which is the beautiful oil. So a blend of those 4 oils are needed for red people in different locations. So they may need flax for their skin, let's say, hemp for their brain, olive for their genitals, and pumpkin for their joints. Okay? So in other words, not everywhere in the body needs the same fatty acids. So this is a composite of 4 different oils for the 4 different fatty acids, or the fatty acids contained in those 4 oils for a red person.
End of chapter 7.